Hello students, welcome to another lecture Understanding Biology chapter 29. Today we are going to learn about roots. At the end of this lecture you should be able to describe the four regions of a typical root, describe the functions of modified roots. Roots anchors the rest of the plant body to the soil and help to uptake water and other nutrients from the soil. Roots, in compared to stems, have simpler pattern of organization and development. There are four regions of development in roots. These include root cap, zone of cell division, elongation, and maturation. For last three zones, the boundaries are not clearly defined. So you can see all four developmental zones in this picture. You can see the root cap. Next to the root cap, you see the zone of cell division. And next to the zone of cell division, you see zone of elongation. And then lastly, zone of maturation. So one thing to notice is that for last three zones, such as zone of cell division, elongation, and maturation, the boundaries are not clearly defined. When cells of root, typically when you look at the cap of the root, it actually contain root apical meristem. This root apical meristem divide and then these daughter cells can have two fates. Daughter cells that end up in the tip will become the root cap cells, while the other daughter cells that are on the non-tip end will pass through these three zones of differentiation before they actually become mature. Functionally, root cap actually protect the tip of the root, which contain in the most important part the root apical meristem. They also function in perception of gravity when root is actually growing. And root apical meristem contain two types of cells. They are called inner columella cells as well as outer lateral root cap cells. So anytime you uproot a plant, you look at the tip of the root actually is where the most important part of the root is located, which is called the root apical meristem. And it is also very important that how these roots are able to perceive the gravity. It is actually done by these columella cells, because columella cells actually have these specialized amyloplast. These amyloplast actually are having something to do with the perception of gravity, because they are collected on the side of the cell facing the pull of gravity. So they help the root tip to perceive the gravity to go downward on earth. Now you know that the root apical meristem is located in the center of the root tip. Next zone toward the edge of the meristem is called zone of cell division. And cells derived from the apical meristem, these apical meristem daughter cells divide rapidly in this zone to subdivide into three primary tissues, the protoderm, procambium, and ground meristem. So all the daughter cells that are dividing or divided from the root apical meristem will subgroup into or subdivided into three primary tissues in the cell division zone called protoderm, procambium, and ground meristem. So these genes that are involved in regulating the patterning of these tissues is actually happening in the zone of cell division, and most of the cells are still cuboidal in shape with a small vacuole, proportionally large centrally located nuclei. These cells will not show any anatomical or morphological expression until they reach the zone of maturation, but the genes are actually expressed in the cell division zone itself. So evidence are actually there are genes that are involved in like patterning of what the mature cell type these apical meristem cells will become is actually activated in zone of cell division. So we actually learned many genes that control this patterning using Arabidopsis. So we have two examples. Here are two genes that are responsible for the regulation of patterning of these tissue systems. So the gene werewolf, or we call it short in WER, is actually required for the patterning of the root hair in root epidermal cells. So plants with a mutation will develop excess root hair in non-hair epidermal cells. So this is how we learned that the WER gene is actually involved in root hair development. So we have another gene that is called SCR, or scarecrow, which is necessary for the ground cell differentiation. Ground meristem cells undergo an asymmetrical cell division that give rise to two cylindrical cells. So one is actually outer cell layer become the ground tissue, and they serve as a storage, and inner cell layer forms the endodermis. So it is very important to have this gene functioning for differentiation of endodermal and ground cells. So now we are done talking about zone of cell division. Let's move into the next zone, which is zone of elongation. Zone of elongation, basically through this zone, cells produced by primary meristem will become elongated. They contain small vacuoles that merge and grow into larger vacuoles that occupy about 90% of the volume of the cell. This is actually metabolically efficient for the cell to have bulk of the volume to be filled with vacuole than cytoplasm, because cytoplasm is expensive to make when growth is actually very rapid. So no more further increase in the cell size above this zone, only increase in girth in mature part of the root. So the last zone in root development is called zone of maturation. In this zone, elongated cell become differentiated into specific cell types. Cells of the root surface mature into epidermal cells, which have very thin cuticle and include both root hair and non-root hair cells. Parenchyma cells produced by the ground meristem called cortex, they store food. Inner boundary of the cortex differentiate into endodermis that contain Casparian strips. Endodermal primary walls contain suberin that is not permeable to water. These stripes block the transportation of water, creating selective permeability, which is very important for plants. All tissue interior to the endodermis is collectively called stele. Immediately adjacent to the interior of the endodermis is a layer of parenchyma cells known as pericycle. Pericycle give rise to lateral roots or lateral meristems in eudicots. So let's look into picture of a cross section of a zone of maturation. So you can see the cortex in outermost. Then we have endodermal cells with Casparian stripes. We have pericycle, then xylem and phloem, which is vascular bundles. So it is very important that you know how to differentiate between monocots and dicot roots. The tissue arrangement in monocot and dicot roots are different, so it is very easy to differentiately identify them. So water conducting cells of the primary cell xylem is located in the core, in the center of a young eudicot roots. In a cross section, the central core of the primary xylem actually looks like a star. So if you see a star in the center, then it is a eudicot root, cross section of a eudicot root. On the other hand, the primary xylem are actually in distinctive vascular bundles arranged in a ring in monocot roots. Um, in the center of the monocot root, you can find the pith. It's a type of parenchyma cell. So if you see vascular bundles arranged in a ring, and then the pith in the center, it is actually a cross section of a monocot leaf, monocot root. In both monocots and dicot, phloem is located as a discrete groups of cells adjacent to the xylem. In eudicot, part of the pericycle and the parenchyma cells between phloem and xylem become the root vascular cambium, which will start producing secondary xylem into the interior and secondary phloem into the exterior. Most plants produce either taproot or fibrous root system. Actually, taproot contain a single large root with the small branching roots, while fibrous roots contain many smaller roots. Some plants produce root modification with a specific function in addition to anchorage and absorption. Not all roots produced by pre-existing roots. Any root arises along the stem or other places other than the root of the plant is called adventitious roots. For an example, climbing plant ivy produce root from their stems. These anchor the stem and help the tree to stand. So these are called adventitious roots. Now we are looking at different kinds of modified roots. And here are some examples for modified roots. The prop roots. Some monocots actually produce thick adventitious roots from the lower part of the stem, such as cone. They actually brace the plant against the wind and help them to keep standing. We have aerial roots. If you ever grew an orchid at your house, these epiphytic orchids root extended into the air that help to obtain water from air. We have pneumatophores. Pneumatophores are actually produced in plants that grow in swamps and have a spongy outgrowth that facilitate uptake of oxygen. We have contractile roots. These roots of lilies and dandelions have contractile roots that pull the plant deeper in the soil each year until they reach in an area relatively stable temperature. And we have parasitic roots such as certain plants such as cuscuta that lack chlorophylls produce these peg-like roots called haustoria. They penetrate the host plant and absorb nutrition and become a parasite for that host plant. And we have food storage roots. Xylem of branch roots of sweet potatoes produce extra parenchyma to storage large amount of carbohydrates. We have water storage roots. Some members of family Cucurbitaceae produce these water storage roots that store water. We have buttress roots. Fig and other tropical plants have these buttress roots that provide stability. So let's look into some pictures. So here is cone that has these prop roots and orchids that have these aerial roots. Pneumatophores in plants grown in like swamps, and then we also have these food storage roots, and then lastly we have these buttress roots. So this is the end of the lecture. In summary, the root cap protects the root apical meristem and helps sensing gravity. New daughter cells of apical meristem divide rapidly in zone of cell division, and genes responsible for patterning of these cells into tissue system is activated in the zone of cell division. In zone of elongation, divided cells get elongated and filled with large vacuole. Elongated cells become differentiated into specific cell types in zone of maturation. Arrangement of tissues in monocots and eudicots are different. You should be able to identify the cross sections. Modified roots allow plants to carry out many additional functions. I hope you like this lecture. Until we meet with another lecture, have a good day. Thank you.